Good morning, everybody. Okay, where were we? I do believe we were here. Let's see. Monday again. Can't hear me? Might be just because I'm talking quietly. How about now? Oh, Isaiah can hear me. All right, Isaiah, can you tell everybody else what I'm saying? Berenice, I don't know what's going on your end. Okay, cool. Great. Yay, awesome. All right, let's get going on linkage. So, quick recap, because this is a tricky topic. Independent assortment. What you get for one allele has nothing to do with what you get for another. What you get for one gene, or which allele you get for one gene, has nothing to do with what you get for another, right? So essentially what that is saying is that if you're looking at two particular genes, those two genes are inherited independently of each other, right? They're not tied together in any way. So obviously we have, oh, I'm sorry. Yeah. I need my coffee. Should have coffee before class. Be a good idea. Normally when I ride into work, I do, because I get to work and I have a cup of coffee and I feel better, but I've just been putting the washing out instead. So it's not quite as invigorating as a cup of coffee. So the end result of those two genes acting independently or being inherited independently is if you're heterozygous for both genes, you have equal possibilities of alleles in your gametes, right? And so therefore the inheritance in your children is random, right? Essentially which allele that they will get. Now, we have loads of genes on every chromosome. Varies from over 1,000 to a couple of hundred. And so clearly, not all genes will be inherited independently. Now, kind of going on from that, even if genes are on the same chromosome, they can still be inherited independently if they're far enough apart for recombination to occur between them. And so this is the big, big, big deal of linkage and recombination. Right. Let me tie this up. And that is recombination frequency is proportional to how far apart two different loci or genes are. Further apart they are, the more likely recombination will occur. The closer they are, the less likely they will occur because recombination is random, not RNA-dom, random. So you have a small distance, the random likelihood of recombination occurring between those 2 points is small. If you have a really big distance, the random probability of recombination happening between those 2 points is very high. And you can get gene far enough away that recombination will always occur between them. And essentially, if that happens, you'll get 4 different possible chromosomes, but you'll get the same proportion of allele combinations, right? As you would if those genes were on separate chromosomes, right? So if you look at, come on, this one, you're going to have a 1-to-1-to-1 ratio of these 4 different allele combinations. If recombination always occurs, you will have a 1-to-1-to-1-to-1 ratio of these 4 chromosomes, again, representing allele combinations. Right. If that's so, those 2 genes are sought independently and are not linked. We'll get into what linkage is in a second. Okay. Does that make sense? I've got to make sure, because this is the foundational concept behind linkage. If you don't get it, it's going to be gibberish. If that didn't make sense to anybody, let me know now. All right, and I'll figure out the sticking point. What's the sticking point, Infinity? Give me some idea about what isn't working for you. And don't be shy. I don't mind. I'm not going to take offense. So what's the sticking point? Okay, so do you get the fact that recombination is random? Oh, why they're not linked? Okay, so I mean, the easiest way of kind of thinking about it, in a sense, I always use football. I'd use rugby actually if I was in the UK, probably. But you know, in American football, when they're doing a drop kick goal to those big posts at the end, whatever those are called, right? Imagine if you blindfold, what is it, Tom Grady or something? I don't know. That guy that sounds like Kermit the Frog. Can't remember what he's called. Just kind of like blindfold them, right? Right, spin them around a few times, point them in the general direction of the end of the field. Now, if you get them to kick a ball, just by random chance, sometimes it will go through those goalposts, right? Other random times, you know, obviously depending on their spatial orientation, it won't. Does that make sense? Like this kind of concept of distance and randomness. Right. So now imagine that those goalposts are the same width as the field. Yeah, which would be nice, right? If you're, you know, the one taking the kick. Essentially, unless you're completely useless and can't kick a ball, right, which presumably they're not, given that they're in NFL and they get paid to lose money. If they're as wide as the field, you're pretty much guaranteed that every time they kick a ball, it's going to go between the posts, right? Doesn't matter how skillful they are, it's just going to happen. It's just random chance. So the goals are fully far apart. Ball's always going to go through. And if you think about the ball is recombination basically, and the goalposts are the 2 genes. Now, if one were cruel, how about, yes, Stephanie, they'll always recombine. And it's still random where that recombination occurs between them, just as, you know, if you kick a ball, it could be like over there or over there or over there, just totally randomly. That actually doesn't matter where the recombination occurs, it's just that it always occurs between those 2 points. That's kind of the key. So now take those two goalposts, I don't know, put them a bit less than a ball width, like one of the American football, like oblong things, right? Put them a little bit less than like a ball width apart, right? And now do the same thing, you know, blindfold Kermit, spin him around, point him in the direction of the end of the field. What's the likelihood that even on a great day, even actually with his eyes open, he would be able to get that ball through it? Well, it's very small. Actually, it's effectively zero, right? Because there's no way for that ball to get through those. The hole is narrow in the ball, right? So still the ball will go all different places, right? Randomly, but you'll never see that recombination occur between those 2 points, right? Because they're just too close together for random chance to occur, for that random act of recombination to occur. And that's the central principle behind linkage. The likelihood of recombination occurring somewhere, it doesn't actually matter where, but somewhere between 2 points, increases as those 2 points get further apart. And at some point, distance, it will always occur between those 2 points. And if it always occurs, you'll always get an equal proportion of allele combinations. So if 2 genes or loci are far enough apart, you will always get an equal, I don't know, ratio, I guess, of the different allele combinations. Basically, as if they're on separate chromosomes. I mean, they're not because they're on the same chromosome, but they're behaving as if they are. Does that make sense? Does that help, Infinity? Cool, that's good. Sometimes it just takes thinking about it in a different way, or using a very bad American football analogy. Now, those chromosomes are different, right? So if you look at the bottom here, recombination equals 2 parental chromosomes, which are essentially the outside chromatids in a pair, and 2 recombinant chromosomes, right? Which is what you see down here. Okay, so everybody's got that good, because this is like totally important. That's why I want to spend a bunch of time on it. And thank you for the question though, Infinity, that helped. Okay, so if there's a way kind of get you thinking about it. If the 2 genes are so close together that recombination never occurs between them, right? In which, in a sense, if you think about it, this is like the Y chromosome in human males, right? What we talked about, the haplotype Genghis Khan and stuff. So if they're so close together that recombination never occurs between them, what ratio of parental to recombinant chromosomes you get. Right, this is the same as the analogy of the goalposts being essentially touching right next to each other. One-to-one what, Stephanie? The parental to recombinants? Or the parental to parentals? So, yes, exactly. So if we go back up here, right, we have a 1-to-1 to 1-to-1 ratio. So another way of typing out what Stephanie put, she's completely correct, but another way of putting it, which makes it a little clearer, maybe for some people, is if two genes are unlinked or sort independently, you get a 1-to-1 to 1-to-1 ratio. If they are completely linked, so no recombination occurs between them, you get a 1-to-0 to 0-to-1 ratio, i.e. no recombinants and only parentals. So the key characteristic of linkage, my typing sucks this morning, must be a lack of coffee. Characteristic of linkage is an excess of parentals. Or a lack or reduction of recombinants. Because essentially, if two genes are linked, sometimes there will be no recombination between them. Now, this isn't that recombination is not occurring. Recombination always occurs. But if they're close enough together, just like with the whole goalpost analogy, right? Recombination could be happening outside of them. So you never see it, right? So don't kind of confuse this with recombination occurring or not occurring. It always occurs. It's whether or not it occurs between the two different things that you're interested in, the two things that you can see, right? In terms of one phenotype versus the other. And so whenever recombination doesn't occur, you have a reduction in the number of recombinants or an excess of parentals. Does that make sense? Okay, cool. It's just probability at the end of the day. It's very much a mathematical function. What's gross? Oh. I said the dirty word, didn't I? Mathematical. I'm sorry. I know this is biology. Can't get away from it sometimes. Okay, so essentially, so you remember we talked about epistasis, right? Epistasis is where, you know, the phenotype for one locus can mask the phenotype of another or the genotype of the other, right? And so there you get a very different dihybrid cross ratio, F2 cross ratio. It would be like a, what is it, 9 to 7 for negative, or 12 to 3 to 1, I think, for dominant epistasis. So do the same thing with linkage, right? With two genes which are quite closely linked together, and you still get the 4 different phenotype combinations, right? So this is not like epistasis, where one or more of those phenotype combinations is gone because it's masked. You still see the 4 different types of phenotype combinations. So you see the parental ones, which are like purple flowers, long pollen, which is one on the left, and red flowers, round pollen, one on the right, as well as the essentially the recombinants, right? The combination of those two. But, and this is the key point, you see an excess of the parentals, or you see a reduction in the number of recombinants. And so you no longer have a 9 to 3 to 3 to 1 ratio. You have a something ratio, right? Depending on how close those genes are together. So wherever you see a lack of the non-parentals, the recombinants, linkage is what is causing that. That's the reason. Okay, so again, key point, recombination frequency is proportional to distance. Further apart, higher the recombination frequency, getting up to 100% between 2 points. The closer, the lower the frequency. Getting down to 0%, actually, if 2 genes are completely linked. And somewhere in between 100% and 0%, you will get a reduction in the number of recombinants, because recombination won't always occur between those 2 points. It's all about distance, and we'll actually get back to this. Oh, I'm sorry. Didn't get a good night's sleep. We'll come back to this when it comes to using this as a tool to calculate genetic distance or map distance, to actually figure out how far apart 2 genes are. And this essentially is the whole point of the fly linkage project. Right? That's what you will be doing experimentally in the lab with your flies. Okay, just going to check schedule for a second, see what we're at time-wise. Pardon me. All right. Any questions? Again, want to take this a little steady because we're going to hit a whole ton of math in a little bit. I want to make sure you understand the reasoning behind it. So another way of thinking about this is kind of an old-fashioned way of talking about linkage, is that if 2 genes are linked, you find alleles traveling together. So I can spell alleles. So if you get the dominant allele for flower color, I guess, you're also very likely to get the dominant allele for pollen shape, using the example of the thing on slide 7. So if you get 1, it's almost like you don't have to worry about, what else you're getting for the other gene, because you'll most likely get the same allele for the other one, not the same allele, you know, the one that was present with that one in the parent. And likewise, if you see plants with round pollen, almost certainly they're going to have red flowers as well, right? Because those two genes, those alleles, the recessive or dominant or whatever, right? Those travel together. So if you get one, you're probably going to get the other one. And the probably bit depends on distance. So if they're very close together, you're pretty much certainly going to get the matching allele, right? And I've actually used this many times, actually. Probably be using it soon when I set up a genetic cross that I need to do for my work. But you can actually use what are called genetic markers, right? So these are genes that when mutated give a very observable phenotype. And so if you manage to get that mutant allele together with something else you're interested in that's very closely linked, you don't actually need to look for the thing that you want. You just look for your marker. And if you have your marker, you also have your gene or allele of interest, right? So it's actually used as a technique as well, something I've used a bunch. So another key point, and this is going to come to how we talk about this as well, and it's extraordinarily important when we talk about linkage, is what's called linkage phase. And so there are two words that matter here. Oh, I'm sorry. I'm not bored, I'm just tired. Cis and trans linkage. So to be able to see linkage, that organism or whatever has to be heterozygous, right? Because otherwise you'll never see it. You'll get either a dominant and dominant or a dominant and dominant, right? You know, you have to have the potential for different allele combinations in the first place just to be able to see linkage occurring. That's kind of the point of when I said, you know, recombination can occur anywhere along the chromosome. Just because it's not occurring between your 2 points doesn't mean it's not occurring. It's just occurring between points that you can't see the consequences of that. Sounds like the puppy's found one of the cats. There's grumbling and growling and hissing upstairs. She hasn't yet learned that they don't want to be her friends. Anyway, so both are heterozygous for both genes. So she comes down. She's getting big. But which way round the alleles are matters. And so cis linkage, both dominant alleles are on the same chromosome. Both recessive alleles are on the same chromosome. So same allele type, I guess, on the same chromosome. Hey, Pepper. She's not coming down. She likes being with her mama. She's a real mama's dog. Translinkage, they're on different chromosomes. Well, not different chromosomes, you get different combinations. Oh, got the other dog instead. Hello, girl. Up. So dominant allele for one chromosome. Oh, got my older pup with me instead. Yeah, she's probably enjoying a bit of peace and quiet. Absence of pestering. Right, and the reason why this matters so much is because that will inform on what are going to be the parental combinations. So essentially what you're looking for in the outcome of the cross. So if those two genes are in cis linkage, then the parentals are going to be dominant and recessive. So the recombinants are going to be the mix, right? What you don't see in the parents. Dominant, recessive, dominant. You'll always get the mirror image one of the other, right? That's just how it works. In trans configuration, the recombinants are now dominant, recessive, like you see on the bottom right here. So it matters a great deal what linkage phase these two genes are in. And that's something that when I ask you the questions in class, either I'll ask you to figure that out, or I'll give you that information if you need that to figure out something else. So it's an important part of the deal. And also and this is the kind of notation that I'll be using in lab. So some of you have seen this already. Some of you may not have. So this is actually flipped around the other way. So cis configuration is on the right. So typically we use, so this is a bit of a departure from like the old school Mendelian stuff, right? We don't use upper and lowercase letters, right? Because it gets confusing, right? You know, if they look very similar. Instead, what we use is dominant allele is a plus. And that's called the wild type. Allele. The recessive has a letter, right? And so W here is the recessive allele for W gene, whatever that might be, Y in the case of Drosophila. And so when we write it, essentially the top line is one chromosome, the bottom line is another, right? And so the vertical bits, right? So essentially, come on. Don't want no shape fill. Right, so that there is one gene. Come on. There you go. This here is a different gene. So you can see this is the same, they're heterozygous, right? Both genes are heterozygous, but which chromosome has which allele is important. Okay, we're good so far? This is something that you need to practice a lot as well, just like chi-square crosses, stuff like that. So you've already seen examples of the kinds of questions you can get that make you use these mathematical skills. You'll get something similar for the next exam. Okay, so that's all the theory and the basics, right? That's the concepts behind what's going on. Pardon me. Now we can use this to figure out how far apart these genes are. Basically, because if recombination frequency equals distance, then the percent recombination equals distance. So all you need to do to figure out distance, at least map distance, and map and physical distance aren't exactly the same. We'll get into reasons why. Generally speaking, they are, right? All you need to do is work out the percentage of recombination by putting the number of recombinants or percent recombinants over the number of offspring in total. So map distance equals number of recombinant offspring divided by total offspring. Times 100. So here, the distance is you add all of these together, right? So for this M, yeah, for wire miniature wings, map distance equals, what's the total there? 216 divided by 644 times 100 equals, what does it tell me? 33.5%. And recombination frequency equals, the unit is centimorgans, named after Thomas Hunt Morgan. Actually, his mentee, his postdoc, guy called Stuyvesant, I think is how you pronounce it, coined that term. Okay, so that's kind of the, let's not worry about that for now. Okay, let's do a test, right? You gonna help out, Luna? Oh, let's get this straight. There we go. So this is called a two-point cross. Let's say we have and this isn't going to be a dihybrid cross. This is going to be a this is actually a back cross, which is exactly what we do in the lab. Oops. And the reason is, oh, sorry, girl, makes it a little bit easier to figure out if you do it as a back cross rather than a dihybrid cross. So we have, what could we use? Yellow and I think they're called diminutive males. Right, those are the two genes that we're looking at. And we get these different combinations. And we have Just making up some numbers. Can't actually remember off the top of my head what the map distance is between these two. They're pretty close. Okay, so first question is, can you actually see what I'm drawing out? Let's stop this. There you go. First question is linkage occurring? Are these two genes linked? Yeah, actually, you could just have left up your original answer, right? Would have been the same. Okay, so these two genes are linked. How do you know that they're linked? What's the key characteristic you're looking for? Yes, exactly. Because I didn't even tell you what the parents were, right? Simply that you have more of these. Oh, that's a sad looking red pen. That is a very sad red pen. I'm going to have to snag another one from work. Goes from red to pink. Can you read that okay? Hopefully you can. If not, I'll write it out. Need new red pen. Oh, here's a red pen. How about that? Right. And so these are the recombinants. Okay, so we've established that linkage is occurring because we have more of these than we have of these. If there is no linkage, we should have an equal number of the different phenotype combinations. Okay, so we've got that straight. How do we work out the distance between these two genes? First step. What's the total? Let's see if I can do that off the top of my head. That is 260, 302, 532, I think. Please feel free to check my math. I think that's right. 60, yeah. Oh, 538. Oh, wait. Oh, yeah, I'm sorry. Cool, thank you for that. It's always good to have people check your math, right? I have the luxury that I can have people do that for me. Okay, so recombination frequency is what? How do you work it out? What are we dividing by what? Yeah, we've got to add the recombinants together and then divide by the total. So I'm going to take a risk, and I think that's 80. So we have 80 recombinants divided by 538. And all of that times 100, which equals what? Oh, yeah, fast. Okay, so that's 14.87%. And that is exactly the same as 14.87 centimorgans. And that's how you work out distance between two genes. It's really quite straightforward. You know, it's as long as you can like add things up. I should use a calculator. That would have been a whole bunch more reliable. Now let's do another one because do it quickly before the end of class so you can. So here you go. There's one that I was actually using this earlier. So these two genes in C. elegans, this is a marker I use for this one. And if you were to do a cross between those two, you would get the following outcomes. So this is called UNC because it's uncoordinated. It's really funny. It doesn't go backwards. If you poke its nose, it will only go forwards, which is pretty weird. So you do this again, we're going to get, actually, let's do some big numbers. What's the map distance between let 23 and UNC4? Again, can't actually remember exactly how close it is, but it's really, 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 really close. Okay. You have 1 minute, people. First, add up all of the offspring, which gives us what? Be like 1559, 1564, I think. Oh, 2564. Yeah, my mathematical brain is just not working today. Okay. So the combination frequency is what? How many recombinants do we have? 5. That one's easy, right? Even I could do that. So recombination frequency is what divided by what? Going to be 5 divided by times 100 equals, I'm not even going to try and guess that. Is that times 100 already? I think it is, isn't it? Awesome. Thank you, Bianca. Percent equals 0.2 centimorgans. So these two genes are very tightly linked. And I know this for a fact because I've done a whole bunch of work and they, unless you really, 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 really push it, you'll never see recombination occurring between these two genes. Okay, so that's enough for today. I need to get myself some coffee so I can function for the rest of the day. What we're going to do on Wednesday is we're going to look at 3-point crosses. So we're going to look at 3 genes, right? And there's a bunch of reasons for that, because obviously you're going to be doing this kind of experiment with more than 2 genes if you want to map a chromosome. So have a look through that. That actually gets a little bit, well, even more kind of, you know, so-so. So have a look through the PowerPoint, and then we'll tackle that on Wednesday. That's actually the bit that people struggle with the most. This is relatively straightforward. It's not too tough. 3-point crosses are a little trickier. Okay. Cool. Thanks very much, everybody. It's been a blast. And I'll see you either tomorrow or on Wednesday. Bye bye.